Welcome to Introduction to GenAI in Drug Discovery and what you can expect from Makya. In this module, we are diving into how generative AI is actually being used today in drug discovery and what Makya brings to the table that isn't just more noise. Generative AI is starting to transform drug discovery, but it's important to separate the hype from the real impact. Traditionally, discovering a new drug takes around 12 years and costs nearly 3 billion. Why? Because you're essentially searching for a viable molecule in a chemical space of about 10 to the power of 60 possible compounds. Now, here's where generative AI comes in. By mining massive datasets and probing just a tiny slice of that vast chemical space, AI help us collapse that timeline, potentially down to a few years or even months in some cases. But let's be clear, we are not eliminating the complexity, we are just managing it better. The workflow breaks into three key phases. Design. AI models can now generate novel molecules in a matter of hours, not weeks or months. But generating molecules isn't the hard part anymore. The real challenge is making sure those molecules are worth the effort. Optimize. This is where feedback loops matter. AI can rapidly refine molecules using real-time data, a huge leap from traditional iterative wet lab work. But again, the quality of the data determines the quality of the optimization. Validate. Here's the more pragmatic part. Prioritizing compounds that are not just promising on paper, but actually synthesizable and low risk. This step is often where AI initiative hit a wall. Chemistry is difficult. Models don't always account for that, but the best platforms are getting smarter at flagging red flags. So while generative AI won't replace the scientific methods, it's changing the game, shifting us from a random screening to a rational exploration. In drug discovery, multiple approaches are used to identify new candidate molecules. Traditional methods like virtual screening operate within the bounds of known chemical space. This inherently limits novelty and often constrains success rates, all while incurring high computational costs. One clear advantage of virtual screening, however, is that the compounds are typically synthetically accessible, a crucial factor in real-world feasibility. In contrast, GenAI offered access to a much broader chemical space, unlocking the potential for novel structure tailored to specific project objectives. It's faster and computational cheaper, but that comes at a cost. Most of the generated compounds tend to be difficult to synthesize, which significantly increases the cost and time required for chemical synthesis and experimental validation. At Ictus, we focus to bridge the best of both approaches. How do we do this? Our unique technology explores the chemical space by using commercially available building blocks and chemical reactions, unlike other GenAI that uses text representation of molecules lacking of any chemical insight. This enables the generation of novel chemical structure which are optimized to meet your project objectives and which are easily synthesizable for rapid test and analysis. In recent years, we've seen an explosion of generative models for molecular design. Over 40 approaches reviewed in 2024 Nature study. But there's a critical flaw across most of them. They rely on molecular representation like smiles or graph without considering the reality of chemistry. They often ignore whether the building blocks are actually available, whether non-synthetic route exists, and whether the molecules are even synthetically feasible. This is where Ictus GenAI stands apart. It's chemistry first. The model is built with synthesis in mind. using available building blocks, known reactions, and prioritizing feasible molecules by design. So while other models might generate novel molecules, they are often dead ends in the lab. ICTOS focuses on generating what matters, compounds that are not just optimized for targets, but can actually be made and tested fast. Now that we cover how ICTOS GenAI can drive novelty while considering feasibility, let's look at how we actually design optimized molecules that meet real project goals. This is where chemistry guided de novo design comes in, a process driven by the target product profile. That means we are not just generating molecules randomly. We are designing with a purpose, incorporating data like the target crystal structure, known ligands, chemical space constraint, predictive models, and even reactivity rules. At the core of the system is a reinforcement learning engine integrating 3D ligand-based designs, 3D structure-based insights, predictive models, external APIs, and similarity. This closed loop in silico DMTA cycle enables rapid iteration towards synthesizable molecules that actually match the target product profile. We are not generating molecules for the sake of novelty, we are optimizing them towards the actual needs of the project while ensuring they are synthetically viable from the start. Building on the previous concept, once we define the target product profile, the real work begins. We start with our generative AI, which designs novel chemical structure. But this isn't unconstrained generation. It's grounded in real-world chemistry. The AI pulls from a library of over 1 million commercially available building blocks and more than 2,000 well-established organic reactions. In other words, every molecule proposed is, by design, synthetically accessible. Then we enter the optimization loop. These generated molecules are scored using reinforcement learning based on how well they meet the target product profile. This scoring system acts like a reward function, guiding the AI to prioritize more promising candidates in every iteration. The results, a tightly integrated chemistry aware design cycle that doesn't just generate ideas, it delivers optimized, synthesizable molecules that are aligned with real-world project goals. With our generative AI platform, we cover the entire early drug discovery workflow, from hit identification and scaffold hopping to multiparametric optimization and retrosynthesis. It's a fully integrated chemistry-aware system that generates, scores, and selects optimized and synthetic feasible molecules, enabling fast transitions from in silico to in vivo. To wrap it up, our generative AI isn't limited to a single task. It's built to handle a wide range of drug design challenges, from scaffold hopping and hit identification to lead optimization with multiple objectives. We cover the full spectrum, all while ensuring synthetic accessibility, 3D constraints, and multiparametric optimization aligned with your target product profile. This is a modular, flexible platform designed to tackle real-world constraints, not just generate molecules, but deliver viable candidates.